Hello, welcome to Microbiology Practical 1. Today, we are going to learn about materials, safety, and distribution of microorganisms in nature. The objective of this experiment: first, I would like you to know the tools and equipment used in microbiology laboratories. And second, I would like you to be able to indicate the distribution of microorganisms in nature. Before going to work in the laboratory, there are some precautions that you need to know. Please read the instructions in the lab manual and follow them strictly. Then, hand washing is required before arriving and leaving the lab. The use of laboratory gowns, clothes is required. No food and drink is permitted. Keep your workspace free of all unnecessary materials and report spills and any accidents to instructors and clean small spills with care. To conduct microbiological laboratories, certain equipment or tools are required for culturing and sterilization. Therefore, students should know how to use the basic microbiology tools for safety precaution. Tools and equipment that students should know are, for example, first, culture tube or test tube. In microbiology laboratories, a large number of test tubes of various sizes are required to be used for microbial culture media. Tubes come in many sizes. The top opening area of the tube may be covered with cotton plugs, metal cap, plastic cap, rubber stopper, or screw cap. The in vitro culture medium is variable in both agar and both solids. The tube must have a stopper to prevent other microorganisms from contamination. In some operations, a small glass tube measuring 5 to 50 millimeters, which is called Durham tube, may be inserted to collect gas produced by microorganisms in culture tube. Petri dishes. It contains two pieces of the plates, one larger than the other, to cover the other. It can be made of glass or plastic or disposable, to be used for agar medium. Petri dishes have more surface area than that of the culture tubes. The amount of culture media to be contained in each petri dish is approximately 50 milliliters. Any less will cause the food to dry out and lose its properties when placed in an incubator. Bacteriological pipette. In vitro bulk, liquid, or solid inoculation requires a pipette. There are many sizes to suit the type of work. There will be a cotton ball plugged at the end of the pipette to prevent contamination, and the red rubber ball must be used to aspirate a solution when using a pipette. Do not use your mouth. Bacteriological slides. The size of the slides is about 2.5 to 7.5 centimeters. They are used as a support for microscopic specimens. There is also a slide with a concave slide to observe the motility of the microorganisms. Inoculating loop and needle. It is a device necessary to transfer microorganisms from one container to another. They are made of good thermoconductive wire, such as nichrome or platinum. It has soluted handle. The inoculating loop has a loop shape at the end of the vial, coiled in a circle about 1 to 2 millimeters in diameter. As for the needle, it has a straight tip. When used, it must be burned or flamed to red hot vial and let it cool for 10 to 15 seconds before transferring the microorganisms. Forceps is the tool that you normally use to take sample or some small tool for experiment. The forceps should be flamed or sterilized before use. But prior to flaming the forceps, you may need to dip the forceps into 95% alcohol and then flame. Incubator. Incubator is an oven that can adjust the temperature according to the need. It is convenient to use because there is a dial for turning to select the desired temperature. The cabinet has a system to adjust the airflow to make the temperature consistent throughout the area. It is very useful in culturing bacteria, especially those are normal microbiota or pathogen microorganisms, because most of which can grow best in 37 degrees Celsius. Refrigerator. Refrigerator is used to store unneeded culture media, as well as various chemicals and reagents that need to be stored at low temperatures, such as antibiotics, serum, blood, and plasma. It can also be used to keep the microorganisms that need to be stored for long-term studies as a stock culture without further cell division. However, this is except for some bacteria that die easily at low temperatures. Bunsen burner. Bunsen burner is a lamp that uses flue gas to create a flame for sterilization that is attached to certain tools, such as sterilization needles, pipettes, test tube, tweezers, and the most common inoculation tools. Alcohol burner. It is a type of lamp that it uses alcohol as fuel to produce flames. The use of this alcohol lamp is the same as a gas lamp or Bunsen burner. If the laboratory does not have gas, the flame from an alcohol lamp is much less hot than from the gas lamp. Therefore, it takes longer to burn to sterilize. Water bath. Water bath is a container that can adjust the temperature according to the need. It can be used for inoculation in cases of culture in liquid medium or in vitro, and used in the preparation of agar medium, or heat the culture medium to an appropriate temperature for the experiment. Sterilizer. It is used to remove all living things from the culture medium, as well as tools and various equipment. Methods for destroying or eliminating microorganisms and other living things is called sterilization, which has many forms, but the equipment used should be selected to suit the type of work. Here are examples of sterilizers. Autoclaves. Autoclave is an equipment that is used to kill all types of microorganisms, and it is a very popular method for sterilization. The sterilization process is done using heat from boiling steam under pressure and heating at 121 degrees Celsius with the pressure of 15 pounds per square inch for 15 minutes. This sterilizer is commonly used to sterilize surgical instruments, agar media, petri dishes, test tubes, as well as to keep all the medical equipment free of microorganisms. The appearance of the machine is a rectangular or cylindrical metal container with a strong lid. Inside, there is the chamber for packing items that need to be sterilized in the same manner as steaming. There is a space at the bottom of the chamber for filling the water. Hot air oven. Hot air oven is the sterilization cabinet using dry heat at a very high temperatures. The inoculator has an adjustable high temperature, usually at 180 degrees Celsius for 3 hours. This type of tube is suitable for disinfection from heat-resistant materials, which do not deteriorate when exposed to direct high heat, such as glassware. Millipore filter. The filter's pore size is so small that the bacteria cannot pass through it. Therefore, the filter substance is free from bacteria and other organisms except viruses. It is used for thermosensitive substances such as protein, plasma, serum, or antibiotics. When you are working in the microbiology laboratory, one rule that you need to consider is aseptic technique. Aseptic technique is a technique to prevent contamination while working in the lab. As you know that microbes are everywhere, such as on the benchworks, hands, tools, and equipment, even in the air in the laboratory. Therefore, it is necessary to know the technique of microbial transfer that will not allow the contamination with other unwanted microorganisms. The technique is called aseptic technique, which is a technique that prevents unwanted microorganisms from becoming contaminated with the microbes being tested. And here are the processes that you normally use in the lab to prevent contamination of microorganisms. First, you need to wash your hand before working in the lab. And then you need to clean your bench work with 70% alcohol. And when you're working in the lab, you will also need to wear a lab gown and also work close to the flame. And the flame is very important because you will need to use them to sterilize all the tools or equipment that you will be used to transfer your culture. And you also need to practice how to open and close the container and to sterilize them properly. For aseptic technique, this technique also prevents the microbe from entering the environment, which can be dangerous to the experimenters or others as well. As nurses are medical personnel who provide care to patients 24 hours a day. Therefore, they should have an appropriate knowledge and understanding on prevention and control of spreading of the diseases. This includes countering the cycle of the infection process, destroying or reduce the source of pathogens which can cause infection to others. So, the first experiment, I would like you to study the tool and the equipment used in the microbiology laboratory. Let's move on to the first slide. As I have introduced you about the tool and equipment that used in the microbiology laboratories, So here are some examples of materials and equipment that we normally use in the lab. And then you will need to draw a picture of the tools and equipment and briefly describe their characteristic and how to use them. I'm going to send you the paper of all the example of the equipment that I would like you to describe them. Please observe their character and also learn how to use the tools and equipment and record in table 1.1. The tool that I would like you to write down the characteristic and the picture of them are inoculating loop, needle, Bunsen burner, petri dish, culture tubes, pipettes, autoclave, and hot air oven. Then move on to the next experiment. The second experiment for today, it's about microorganisms in nature. In nature, microorganisms have together in a mixed group. However, in the laboratory, we can isolate these microbes into individual types by culturing the microbes on the agar plates. Microbes multiply into colonies, making them visible to naked eye. Each colony contains millions of microbial cells. As you can see from here, after growing some microorganism on the agar plate, you will see several types of microorganism growing. These are examples of microorganisms that are growing, that are contaminated in the air. And this is the colony of the bacteria. And this is the colony of the fungi. For experiment 2, the materials that you will need are nutrient agar or NA agar for 3 plates, normal saline solution for 1 tube, and sterilized cotton swab for 1 stick. Method of experiment. First, you will need to label at the bottom of the plate using permanent pen and do not open the plate or the lid. Then you will need to divide the plate into 5 sections. For this experiment, I'm giving you 3 plates. And for this experiment, you will need to perform as follows. For plate 1, we are going to use it as a control. So you don't need to do anything with this plate, just to test that the agar medium that I prepare for you is free from contamination. For plate number 2, you will need to use this plate to take the sample from the air. For the air sample, you need to keep the plate opened by opening the lid and then leave the plate in the room for 10 minutes. Then put the lid back, and then you need to put the lid on when done, and then put the plate in the incubator. For the third plate, you will need to divide the plate into 5 sections, as I show you in the picture. The first section, we are going to experiment the bench. The microorganism that contaminate on the bench work. And plate number 2, we are going to study the microorganism that it's contaminated on your unwashed finger. And the third section is for washed finger. And for the fourth session, we are going to test the microorganism contamination from the tap water. And the fifth section, we are going to test the contamination of microorganism from corn. For the first section, you need to dip sterilized cotton swab in normal saline solution, then rub onto the benchwork surface. Smear the swab on the NA plate thoroughly and close the lid. Section 2, stamp unwashed thumb onto the plate and then close the lid. Section 3, stamp washed thumb onto the plate, then close the lid. Section 4, collect tap water using inoculating loop, then smear onto the plate. Section 5, place a coin onto the plate using sterilized forceps. Remove the coin and then close the lid. After finishing all four test experiment, you will need to incubate the plates at 35 degrees Celsius for 24 hours by placing the plate upside down. Then, the next day, you will need to count colony numbers and record the size, type, and color observed on each section, and then write down the result on table 1.2. The video of this experiment is showing in the YouTube channel. Please follow the link. Okay, before we end this practical, I have some questions for you to answer. First, from the experiment, in which steps that you need to conduct the experiment using a septic technique? Second question, which sources were microorganisms the most frequent found? Third, describe differences of microorganisms found in washed and unwashed finger. Thank you.